Get going. I'm Christa. I'm a fourth-year graduate student now in bioinformatics, and I mostly study human genetics and computational genetics. Lately, I've been getting into some EHR type research, but for this talk, I'm just going to be talking about cell-free DNA, which has kind of been the first half of my thesis work. I'll tell you a little bit about CelFIE today. I'll talk a little bit about our biological background and motivation for this problem, the method we came up with this. And so actually, even though the first talk was really different in topic, it's going to be an EM method too. So kind of unified in that way. I have some simulations that I'll show you to support our method. And at the end, I'll show some real data results that we're really excited about. So to start with some biology this morning, cell-free DNA is DNA that enters the bloodstream and it comes from dying cells. And so this could be apoptosis, necrosis, and they shed it into the bloodstream and you can analyze these pieces. And so the goal of our work is to be able to get an estimate of what cells are dying throughout the body. And so we want to be able to take a measurement of cell-free DNA in the blood and say some percentage of it comes from esophagus, some percentage of it comes from skeletal muscle. And this could give you just an overall picture of what's going on in this person at that moment in time. However, just if we were just going to take the DNA and sequence it as geneticists tend to do, this wouldn't really give us that much information about the tissue of origin. And so this is just a factor of the fact that every cell type in our body has exactly the same genetic sequence. But you can use methylation. And so methylation is these marks that happen in the context of a CpG site, a cytosine guanine dinucleotide. And each tissue has a different patterning of these methylation marks, and that influences what genes are turned on in that tissue, which helps, influence that tissue's function. And so this is actually a great way of having us have information about what cell types the cell-free DNA is coming from. So in this really simple example, you look at this first CpG site where the dark green circle is it's methylated and the open circle is it's unmethylated. If we have a theoretical person that's only made up of brain, B cell, and muscle, if I saw that CpG site was unmethylated, I would know for sure that it came from a B cell because in brain and muscle, it's always methylated. So this is how we can start disambiguating what cell-free DNA is coming from what tissues. And of course, we're not the first people that have thought about doing kind of the cell type decomposition problem using DNA methylation. Andres Hausmann in like the early 2010s did a lot of work with decomposition along with Elior Rahmani, who was at UCLA. And these works generally, work like this. They measure some sort of methylation, and usually they're doing blood because these studies were really interested in coming up with the proportions of blood cell types as confounders to correct for in an EWAS study. So they'll measure blood on like an Illumina methylation array, and it'll be over CPGs. And then they'll compose a reference panel. And so in this case, it's different types of blood cell types over the same CPGs, generally like measured on the same technology. And they'll perform some kind of optimization to get back an estimate of each cell type in their reference, what percentage is their sample made up of that cell type in the reference. And so this is great, and you could apply this to cell-free DNA, and a paper from a few years ago did do this type of approach. However, there's some limitations of this that really don't address the specifics of cell-free DNA data. And the first one is just a practical one. Methylation chips, the technology that these techniques were using, require a lot of blood. And so in cell-free DNA, which is only present in the DNA, in the blood, as a pretty small level. If you're trying to think about something that would be clinically relevant, we may not want to be asking patients for tubes and tubes of blood to get the amount of DNA that we would need to perform the chip. So we make this trade-off where we're moving to whole genome bisulfite data, which requires less blood. We can have a little bit of lower DNA input, but now our data is a little bit more noisy. So it's lower read count, we're spreading signal across the whole genome. And I think one of the really big limitations of these methods is that there's an inherent bias. If I'm the one selecting the tissues making up my reference panel, my constrained optimization method is only going to estimate those tissues in my reference panel. And it's really likely that there are rare cell types that aren't well characterized, or maybe I missed a few cell types. I don't really know the full biological picture of what's going on in my sample. So this could cause some bias, and I hope to convince you later that it does. So in order to address these, we developed CelFIE, which is an expectation maximization algorithm that tries to decompose cell-free DNA data, but allowing noisy data and allowing these unknown missing cell types. So similar to the methods that I presented before, we're going to have some reference panel that's going to be over cCPGs. And so in our reference panel, you could have this chip data with a methylation array data that other methods use. But now we're also going to allow for a whole genome bisulfite data, which is read data. So it's the number of methylated reads, comma, the number of reads total for that site. And we're going to have it be on different tissues that the cell-free DNA could be coming from. But crucially, our data also allows for this unknown component. So in practicality, it's just like telling the algorithm that there's like a question mark there, and that we want you to estimate that question mark. And our method is really flexible. It allows for an arbitrary number of unknowns. But for this simple example that I'm going to walk you through, I'm going to only focus on one unknown, and we'll just restrict ourselves to whole genome bisulfite sequencing data right now. And so again, like the previous methods, we're going to measure our samples in the same CPGs that our reference panel is over. But this time we can have, 1 to n individuals of cell-free DNA data that we measure in the blood. We're going to perform an optimization, and this optimization is going to be with an expectation maximization to estimate our proportions matrix. And so in this case, now we have all the tissues that are in our reference panel, but we're also having this unknown category. So our EM will help us estimate, say that there's 10% of our cell-free DNA is coming from a tissue not contained in our reference panel, 1% is coming from lung, 3% is coming from heart, et cetera, et cetera. And so to dig into exactly how we're modeling this a little bit more, I'll start with talking about the reference, which is going to be a binomial model that's parameterized by the methylation proportion in a specific CPG in a specific tissue. So we usually get that data from like the ENCODE consortium, which has a nice compendium of reference data. And then it's also a function of how deeply that tissue was measured. So how many reads are covering that specific CPG at that specific tissue. And this helps us kind of naturally account for the fact that there's varying coverage throughout a whole genome sample, and that varying coverage can give us different confidence in our measurements. So for our cell-free DNA input matrix, we're going to parameterize it a little bit differently, where we're going to look at it one read at a time. And so this is just a simple two-dimensional matrix, but since we probably have multiple reads at a given position, this could be thought of better as a multi-dimensional matrix, where each entry is whether that read is methylated or not, and that specific individual at that specific CPG. And so whether that's methylated or not is going to be a Bernoulli function, and that's parameterized by what's the methylation proportion at that CPG in the true tissue of origin for that read. And of course, this is what we don't really know, the tissue of origin for that read, and that's what we're going to try and learn with our expectation maximization algorithm. And so here, we're going to be updating our alpha vector, which is our proportions, as just a function of the reads at that from that person in that CPG, multiplied by the probabilities that it's methylated at that tissue in that specific person at the specific CPG. And so that is what we're going to be learning and updating in our E step. But also interestingly, our model is allowing us to learn the betas, which is the methylation values of the true tissues, the true reference tissues that are in our sample. And this is just a weighted average of the reads that are coming from our reference panel that we know are in, that we measure with the ENCODE course consortium or something. And then the reads that we're attributing to be from that tissue that we're learning from people. So that way we can start to learn the unknown proportions, the unknown methylation proportions as well. And the E step is where we're just trying to learn the origin status for each of the reads. Okay, so that's a brief overview of our model. So now I'll talk about some simulations that we're demonstrating that our model works. And so first, I just do a simple simulation where I have reads that are approximately 10x coverage, which is a very low coverage for a whole genome bisulfite sequencing experiment. Most whole genome bisulfite sequencing experiments aim to be like 30x coverage. And we're going to do it for 1,000 CpG sites, which again is a pretty low number of sites when you consider there are 28 million CpG sites in the entire human genome. And we're going to do it for 10 tissues that are truly in the mixture. And when we do it in this very simple scenario, we do an excellent job at recapitulating the true methylation proportions. So the true proportions are in red, and the blue are what CelFIE is estimating for that tissue. And so we do a pretty good job. And notably, if we look at the betas, so the methylation proportions of the tissues, we do a really good job at learning those as well. But this is a really simple idealized scenario. I had made the claim earlier that if we are likely to have tissues in our cell-free DNA mixture that aren't contained in our reference panel. So what happens then? Is it worth us doing this fancy EM? And I would argue yes. So this simulation is pretty similar, except now we have 10 tissues truly in the reference mixture, truly in the cell-free DNA, but we only have 9 tissues in our reference. So one is missing from our reference. And so that would be this tissue here. And the red dots are the proportions that are truly in the mixture. And you notice that for each of our estimates, when there's a missing tissue, we kind of overshoot it a little bit. It's trying to, the algorithm is saying that there is some sort of signal there, but it doesn't know where to put the signal. So it kind of spreads it over all of the other tissues that are being estimated. And of course, we can't estimate that at all. However, if we do run CelFIE with this unknown tissue that I was trying to convince you is needed, we're able to recapitulate that missing tissue pretty well. And again, for the pretty low depth and the pretty low number of sites. And we can even begin to estimate the methylation values for that missing tissue. So this is the correlation between the true methylation proportions that I simulated the mixture from and the ones that CelFIE is estimating for that unknown. So these simple simulations, oh, sorry, actually I wanted to just note that I told you before too that CelFIE can estimate an arbitrary number of unknowns. But how do we pick the right number of unknowns for our experiment? And so we've found that depending on the number of unknowns we have, and these are at pretty high proportions of the mixture, they're making up 20 or 10%, that we're going to need more people to estimate that unknown proportion, especially with more unknowns. And this is the reason that this is because CelFIE will tend to overfit to the people that are in your study. If you only have one person in your cell-free DNA panel that you're trying to learn about, and you say that it has one unknown, it's going to say, oh, I can do an excellent job at estimating this person's like cell-free DNA by just, fitting a custom unknown of that person. And so we do need more people, but 100 people seems reasonable. So keep that in mind for when I'm showing you like data further on. But yeah, so I showed you pretty simple simulations. Now I want to turn to some more complicated realistic simulations. So I'm going to take real whole genome bisulfite sequencing data, and this is from the ENCODE consortium, and I'm going to partition it into 2 data sets. One set of biological replicates is going to be my reference, and the second set is going to be my input data, so my pseudo cell-free DNA mixtures. And I'm going to make different proportions of this. So I have, a few different mixtures for an arbitrary number of people. And so in this example, I ran CelFIE I ran CelFIE on these different mixtures, and I have, these real tissues in the mixture, and the red dots again are the true proportions. And you can see that CelFIE does a pretty good job at estimating the true proportions in these more complicated, challenging data. And that we can even estimate a pretty large and substantial unknown decently well. So in this case, T cell is missing, and we're doing a good job of estimating it. And in this case, we have small intestine in T cell missing. And again, we're doing a pretty good job. So this was for 100 individuals. But I'm sure you're like not 100% convinced by that simulations experiment. So I'll try and show you some real data, which hopefully will be a little more convincing. So first, we decided to do cell-free decomposition on pregnant women because this offered a really nice natural experiment for us, since we know that pregnant women will have cell-free DNA coming from the placenta. And hopefully, if you're not pregnant, you won't have a placenta, so there should be no DNA originating from that. So this is a pretty small study, but we're estimating about 20 different tissues, and we're going to estimate one unknown. And we find that indeed, we estimate a significantly higher proportion of placenta coming from pregnant women, and almost none coming from not pregnant women. So this really gave us confidence that our method was working and worked as we would expect. And this is interesting, and we can also use this to glean some insights about biology. So in this example, I could break up the pregnant women by trimester. We only had trimester 1 and trimester 2 in our data, but we can see we estimate a significantly higher amount of placental cell-free DNA in the second trimester women, which makes sense because we would expect the placenta to be growing so it can support the baby. And so future work that we're interested in is, deconvolving things about pregnancy risk, like is there a way we can predict gestational diabetes from pregnant women based on their cell-free DNA profiles? But our final ultimate application of this work was to ALS patients. And so ALS is a really interesting neurodegeneration disease because it doesn't have a biomarker right now. It's diagnosed in general by people going to a clinician and they're ruling out anything else that the ALS could look like. And so it's generally a pretty arduous process, and there's not a good way to kind of quantify people's ALS state. So we thought that this would be a really good candidate for using cell-free DNA and using this as a quantification, since there's this hallmark tissue degeneration that characterizes ALS. In healthy skeletal muscle, we have motor neurons, they attach to our muscles, they tell us to do something. But in patients that have ALS, the motor neuron degenerates, and that causes the skeletal muscle to atrophy. So this was kind of the hypothesis that we had going into this analysis, was that we could observe increased cell-free DNA coming from skeletal muscle. So with our collaborators at the University of Queensland, we came up with a few different data sets. So we have a discovery data set that we're going to apply CelFIE to, and then a secondary validation data set. And right off the bat, we were just looking at cell-free DNA concentration. So is there even any difference in the cell-free DNA in ALS patients? And we found that there was significantly higher cell-free DNA coming from ALS patients. And so this really drove us to ask what's driving this difference between these patients. What cell types exactly are driving this increased amount of cell-free DNA? And so we ran CelFIE for our discovery cohort, which ended up being about 16 ALS patients and 16 controls. Or sorry, this is for our all of our full cohort. And we ran it for 25 tissues or so. And we see, it's a little hard to tell from here, but we see an elevated skeletal muscle component. And so yeah, here's our discovery cohort. And you can see significantly higher proportions of skeletal muscle in our ALS patients. So exactly what we expected. And so when we applied this to our validation cohort, we see the same signal. And when we ran like a meta-analysis of all samples, the signal was still there. So this was really exciting results. It's, to the best of our knowledge, the first time people have used cell-free DNA in the context of neurodegeneration and measuring kind of a quantitative biomarker for ALS. However, we still have a lot of work to do to validate and, kind of hone in on these results. So one aspect of our future work is just to fine tune this algorithm. So there's a lot of things that we could adapt to make the algorithm more accurate. One thing is that cell-free DNA reads will likely cover more than one CpG. So right now we're modeling one methylation site as independent from the next one. But in reality, there's probably 2 sites close to each other, a few sites close to each other that are all coming from the same tissue. So if we could somehow model the kind of relationship between CpGs on one read, we'll probably get increased power for disambiguating the tissues. Another thing we were interested in is having an expanded reference panel. Right now, I only have a reference panel with healthy tissues, but if we included disease tissues, maybe we could generalize our applications to other disease contexts. Like we were thinking about cancer, if we included some cancerous tissue types versus healthy cell types of that same like tumor origin, could we kind of estimate what proportion is coming from a tumor? And the second thing we're working on with the Pellegrini lab is really developing a clinically useful tool. And so we want to come up with a methylation capture panel, which can capture a select number of sites that we know are informative for tissue status, that give us a lot of information to our algorithm. So that way we're able to analyze the cell-free DNA of patients without kind of the cost and burden of having to do whole genome bisulfite sequencing. And so with that, acknowledgements, the Zaitlen lab, especially Noah and Andy from University of Chicago now, have helped a lot with the development of the algorithm. The Pellegrini lab has helped a lot with the understanding of kind of the biological constraints of the problem, especially Marco and Anella. And then we have collaborators at the University of California, San Francisco and University of Queensland. And so with that, I can take any questions.